Hello and welcome to my channel Omni Bioentrance, and today we will be discussing Maharashtra SET Life Science question paper conducted in the year 2024. This is part 2 of the discussion. If you are new to my channel, please do like, share and subscribe. Coming to the next question. Among the different types of phytoremediation, dash is the process in which plants limit contaminated soil movement and migration. So what is phytoremediation? Here the living plants are used to clean up soil, air, water that have been contaminated with pollutants. That is phytoremediation. What is phytotransformation and phytodegradation? Is the transformation of organic pollutants from soil, sediment or water into a more stable, less hazardous and less mobile form. What is phytoextraction? It is the process by which plants accumulate pollutants in their root, shoot or leaves above the ground. That is phytoextraction. So, answer is option A, that is phytostabilization. That is in which plants limit contaminated soil movement and migration. Then next one. Which of the following proteins helps to maintain and stabilize protein tertiary structure during temperature stress in plants? Defensins are to defend against pathogens and predators. Thionins are antimicrobial peptides found only in plants. Late embryogenesis abundant, LAA, that is help to maintain and stabilize protein tertiary structure during temperature stress. Answer is option C. Enzyme labeled antibodies are not generally used for which of the following techniques? FACS is fluorescence activated cell sorting. It uses fluorescent dyes and flow cytometry to sort cells based on various parameters including size, surface markers and fluorescence intensity. Immunohistochemistry, ELISA and Western blot, they use enzyme labeled antibodies, but FACS doesn't use enzyme labeled antibodies. Next one. In watermelons, to induce seedlessness in triploid population, which of the following strategies is useful? That is, you have to cross a tetraploid with a diploid to produce triploid to induce seedlessness in triploid population in watermelons. Answer is option D. Amplification of DNA fragment by PCR requires primers. Which of the following statements are correct for primers? Primers with hairpin structure formation are not desirable. Yes. Primer sequence should not have long runs greater than 3 of a single nucleotide. Yes. GC content of the primer should be less than 20%. No, it should be between 40 and 60%, because when primers anneal to their target sequence, GC can form 3 hydrogen bonds, whereas AT will form only 2 bonds. So 3 hydrogen bonds are stronger than the 2 hydrogen bonds, and separation of these 3 hydrogen bonds, that is GC bonds, it requires more heat than the AT. So it is recommended to keep GC content between 40 and 60%. Both the forward and reverse primers must have high percentage of base complementarities. If that is the case, then forward primer and reverse primer, they will form hydrogen bonds among themselves, and they will not bind to your target DNA. You will get primer dimers. So the answer here is option C, 1 and 2. When subcultured into fresh medium, a bacterium grows with a lag phase of 2 hours followed by log phase and a stationary phase. If the bacterium was treated with ethidium bromide for 3 hours in all the phases of growth to cure a plasmid, it is more likely to lose the plasmid in So plasmid curing is removal of plasmid from the bacterial cells. And ETBR, it inhibits the replication of the plasmid DNA through intercalation. So at which phase it will lose the plasmid is the logarithmic phase. Answer is option C. The free hydroxyl group present either end of a chemically synthesized oligonucleotide are 5' phosphorylated with ATP by the enzyme polynucleotide kinase. Kinase is for addition of phosphate groups. Then next one. Which of the following is not a fluorophore? Acridine, orange, acrylamide. Acrylamide is not a fluorophore. B. Which of the following best describe principle of Western blotting technique? A means of separation of restriction fragments on the basis of size. No. It is based on attraction to a specific chemical group. No. The protein have a net charge 0 at a particular pH. No. It is an immunoassay technique preceded by electrophoresis. Yes. That is correct. In Western blotting, it separates, detects, identifies one or more proteins in the complex mixture. It involves loading of the protein, then you have got the separation of proteins by molecular weight, then transferring to the membrane, then probing with antibodies. Only the protein to be studied can be specifically bound to this particular primary antibody. Then the primary antibody treated membrane is treated with secondary antibody. Then secondary antibody, it will bind to the primary antibody and indicate the location of primary antibody as well as the location of the protein being studied. So it is an immunoassay technique preceded by electrophoresis. Answer is D. Then which of the following is resulted due to photorespiration reaction catalyzed by oxygenase activity of Rubisco? So it is photorespiration. So it is 3-phosphoglycerate and 2-phosphoglycolate. Answer is option D. Mutation in ACC synthase enzyme will inhibit synthesis of. So ACC synthase, it catalyzes the conversion of S-adenosyl methionine to 1-amino cyclopropane carboxylate or ACC. And it is one of the rate limiting step in ethylene biosynthesis. So if there is a mutation in ACC synthase enzyme, it will inhibit the synthesis of ethylene. Answer is option C. Then, which of the following phytohormone accumulates during systemic acquired resistance in plant? So, in systemic acquired resistance is typically based on the hormone salicylic acid. So, answer here is option A. The hormone auxin was first discovered in, so it was discovered in oat coleoptiles. So, the answer here is option C. How many times more energy gained in aerobic respiration over anaerobic respiration? So, if it is 38 ATP per glucose is produced in aerobic respiration and in anaerobic respiration only 2 ATP per glucose is produced. So, 38 divided by 2 will give you 19, but it is not the answer as per the options. Because some textbook they state that net yield is 36 ATPs in eukaryotes because 2 ATPs are used to power cellular respiration itself in moving 2 NADH molecules into mitochondrion. So 38 minus 2 will be 36 and 36 divided by 2 will be 18. So your answer will be option C. Then, which of the following is not involved in electron transport during photosystem 1 and 2? Ferredoxin, plastocyanin, cytochrome B6F complex, all three are involved, but cytochrome oxidase is not involved. It is the final electron acceptor in the mitochondrial electron transport chain and is required for aerobic ATP production, that is cytochrome oxidase. So, D is not involved in photosystem 1 and 2. Then next one. The treatment of abscisic acid is associated with closure of stomata. Answer is option C. Action potential is generated due to influx of Na positive, efflux of K positive. Yes. Answer is option A. Then hookworm infection can lead to iron deficiency. Answer is option B. What kind of microbes grow best at higher carbon dioxide tension than are normally present in the atmosphere? So it is capnophilic microorganisms. They grow best at higher carbon dioxide tensions. Then next one. Which free-living microorganism occupy a niche to feed primarily on organic detritus from dead organism and unable to adapt to the body of live host? So they are obligate saprobic microorganism. They feed primarily on the organic detritus and dead organisms. Then next one. The DNA of each chromosome occupies a defined volume of the nucleus and only overlap with its immediate neighbors is called as chromosome territories. Answer is option A. Then next one. Amphibians contain 30 times more DNA than human beings. However, human beings are more complex than amphibians. This lack of correlation is called C-value paradox. In C-value paradox, the amount of DNA in the it is the unexpected lack of relationship between the presumed complexity of the organism and its amount of DNA in the organism's genome. That is called as C-value paradox. Which of the following is not true in relation to eukaryotic gene expression? RNA pod 1 synthesizes pre-gamma RNA. It should be a typing error. It should be rRNA. RNA polymerase 2 synthesizes rRNA. Yes. Answer is option B is correct. Which of the following is not true? Okay. B is not true. Pol 2 is involved in mRNA synthesis, also synthesizes long non-coding RNA, small nuclear RNA and microRNA. RNA polymerase 2. Then, the triple helix domain of collagen consists of repeats of the XYZ amino acid. The amino acid present at X position is required in every third position in order for the polypeptide chain is. So, which amino acid is occupying the position X in XYZ? So, it is glycine which is occupying the position X, whereas Y and Z can be proline or hydroxyproline. Then next one. Which DNA binding domain is present in the steroid hormone receptor family of ligand-inducible transcription factors? It is zinc fingers. Answer is option B. Which of the following elongation factor is called as translocase? It is EFG and EF2. EFG in bacteria and EF2 in eukaryotes. Next one. Which of the following post-translational modification is not related with epigenetic regulation? Histone acetylation, yes. Sumoylation, yes. Ubiquitination. Ubiquitination is not related with epigenetic regulation. It is for protein degradation. So C is the answer. The disease which arises because of aberrant splicing is thalassemia. Answer is option D. Identify the enzymes involved in DNA replication for their function. DNA polymerase 1 remove primer and close gaps. Primase, it synthesizes primer. Helicase, it is for unzipping of DNA helix. Gyrase is for supercoiling of DNA. So the answer here is P3Q4R1S2. Answer is option B. Then first statement. The 3' to 5' exonuclease activity of DNA, eukaryotic DNA polymerase operates in reverse direction of DNA synthesis and participate in fidelity of replication. Yes. Statement 2. The ability of DNA polymerase to extend a primer only in 5' 3' direction appears to make replication a complicated process. It is necessary for proofreading of duplicated DNA. That is also correct. So choose the correct answer. 1 is correct, 2 is wrong. Both 1 and 2 are correct. Answer is option C. The next one. Which of the following is a wrong statement in SOS repair system? Lex A shows auto repression of Lex A. So Lex A is a protein. It is written in capital, and Lex A here it is written in small letters. So the protein shows auto repression of the gene. Lex A. Yes, that is correct. Lex A shows repression of Recce gene. Yes, that is also correct. Negative regulation of Lex A blocks SOS repair. Yes, that is not correct. Which is the wrong statement in SOS repair system? C is not correct. Recce shows negative regulation of Lex A. That is also correct, because if Recce is present, it will cause autocatalysis of Lex A. So it causes negative regulation of Lex A. Now when there is negative regulation of Lex A, then it will result in removal of Lex A from the SOS box and the synthesis of SOS repair proteins. So negative regulation of Lex A blocks the SOS repair. It is not blocking the SOS repair. It is activation of the SOS repair. So C is the incorrect statement. Then next one. Which of the following statements about apoptosis is false? Cell shrinks. Chromatin condenses. Cell contents are leaked. Cell contents are not leaked. They are shrinked. So C is the answer. Then which of the following signaling molecules play a key role in the patterning of neural tube along with its anterior posterior axis? So it is WNT4. Answer is option A. If you compare B cell epitope against a T cell epitope, B cell epitope are based more on the primary sequence of protein antigen. No, B cell epitopes are based on the nature conformation of protein antigen. Yes, that is more correct. Not on the primary sequence, but on the nature conformation of protein antigen. B cell epitopes are based more on presentation of, it is not B cell epitope, it should be T cell epitope. B cell epitopes are of continuous epitopes than the B cell epitope. That statement itself is wrong. 2 times B cell epitope have been mentioned. So B is the correct answer. Mark the correct statement for antigenicity of molecule. Antigenicity is the ability of a molecule to provocate immune response in the body. It is not antigenicity, it is immunogenicity, that is the ability of a molecule to provocate immune response in the body. And antigenicity refers to the binding ability of antibody to its antigen. So answer B is more correct. Antigenicity and immunogenicity are synonymous. No. All antigens are immunogens. No. So B is correct. Which of the following molecule act as general adhesive connecting one cell to another and cell to other substrates? It is fibronectin. Answer is option B. Then next one. Formation and migration of epiblast as a sheet of cells during gastrulation in zebrafish requires the expression of. It requires the expression of E cadherins. Answer is option D. Then, which of the following groups of transcription factors is responsible for maintaining the pluripotency of stem cells? It is OCT4, NANONG and SOX2. Escherichia pestis is the causative agent of plague. Answer is D. Pseudohermaphroditism is a condition in which the individual is insensitive to androgen. Answer is option A. In a plant, when the apical cell of the two-celled proembryo divides longitudinally and further the basal cell plays only a minor role or none in the development of the embryo proper, then the embryo is onagrad. Answer is option B. Then next one. Which of the following is necessary for the development of epididymis in mammals? It is testosterone. Answer is option C. Then, all these are direct questions. The mid-blastola transition is the point in the development when transcription of zygotic genes begins. Answer is option D. In the chick limb, the signal for condensation of mesenchymal cells to give rise to cartilage comes from the appearance of N cadherins. Answer is option A. Which of the following is not a property of stem cells? Ability to self-renew. Yes, it is a property. Ability to differentiate into different type of cells. Yes, they are unspecialized cells with cell markers. They are actively dividing cell with cell marker changes. So it can be either C or D. They are not unspecialized cell, they are specialized cells with special cell markers. They are actively dividing cell with cell marker changes. So either C or D could be the answer. Then, which of the following proteins act as a death receptor that triggers the extrinsic pathway of apoptosis? It is FAS. FAS. C. With reference to normal ECG, the T wave is caused by repolarization of apex and outer surface of the ventricles. Answer is option B. Increased muscle activity will increase generation of lactic acid. In this condition, blood coming from the lungs to the muscles would show. So increased muscle activity, it is resulting in increased generation of lactic acid, and in this condition, blood coming from the lungs to the muscles would show increased oxygen release from hemoglobin. Because you require more oxygen when increased muscle activity is there. So answer is option A. Then next one. The capability of vertebrate kidney and produce hypertonic urine is a function of. It is a function of hypertonic urine is a function of loop of Henle. Then, for long distance runner, the muscle needs increased oxygen supply. At the muscle, this is formed by increased and frequent muscle contraction, increased demand made by the muscle, increased squeezing of the muscle, increased accumulation of lactic acid causing demand blood pH. Answer is D. Which of the following heat shock protein has both protein folding and degradation activity? It is HSP70. The sequential order of various stages of fermentation process is inoculation, fermentation, downstream processing and removal of waste. Answer is option. Then we match the following product or processes to the microorganism involved. Biopole, alkaligens eutrophus, biopesticide, Bacillus cepharicis, bioleaching, Thiobacillus ferredoxins, and bioremediation of oil is Pseudomonas putida. So the answer here is option D, L4M3N1O2. In golden rice, the gene phytoin desaturase was obtained from Erwinia euridovira. Answer B. So that's it. Thank you for watching my channel. Please do like, share and subscribe.